These are all the files that we have downloaded in our previous tutorials. So now we will load all these files one by one into R. So first of all, we will get started with loading the .h5 file by using the read 10x reader function in R. So load the .h5 file. So counts read 10x function. So now we will copy the file path of the .h5 file. So copy and we will paste into the code and press the run button. It will take some time. The file has been loaded, and now we will check the structure of that file by using the dim function in R. Counts and run. The output shows that this file has these number of rows which are the actual detected peaks, and these number of columns which are the actual detected cells after filtration. So now we will check the first few rows and columns of that file. So first 12th rows and first 4th columns and run. Now that is the actual structure of that file. The rows show the number of detected peaks and the columns show the number of cells. Each column is represented by a unique barcode. That is a type of sparse matrix, and most of the entries are dots, which means 0, and this means that most of these cells have 0 cutting sites for the TN5 restriction enzyme for that particular peaks. But there are also present some non-zero value, including the 4 and 2. So what does mean by the 4 and 2? The 4 means that this particular cell type has 4 cutting sites for these peaks for TN5 restriction enzyme, and the 2 means that this particular cell type has 2 cutting sites for TN5 restriction enzyme for this peak. Now we will load the .csv file by using the read.csv function in R. So first of all, we will list all the files to copy the file path, and that is the .csv file. So we will load the .csv file. So metadata is equal to read.csv function, and now we will copy the file path. So copy and paste into the code. Header is true. So now so all the row will be now the column, and row dot names is equal to 1, and the column which have the barcodes will now become the row. So press the run button. The file has been loaded. Now we can check the structure of that file. So metadata, we will look into the first 6 rows and first 6 column. Run. So that is the actual structure of that file. This file has some extra information about the cell type, about each cell type, including the chimeric, unmapped, and mitochondrial content. We can also view this file, metadata. So that is the whole structure of that file. We will also use this file later for creating the Seurat object. Now we will create a ChromatinAssay, which is one of the most important step, because normal Seurat object stores the gene expression data, and ChromatinAssay stores the peaks. So first of all, we will list the file and to the local directory, and now we will create a ChromatinAssay. So ChromatinAssay, we will use the create ChromatinAssay function in R to create the ChromatinAssay. So create a ChromatinAssay function, and now we will pass the counts, which is a sparse matrix. So counts, and these will tell the Signac how to name the peaks. For example, we will just look into our previous file. These will tell how to name the peak, including the chromosome number, and the genomic ranges, including the start site and end site. And now we will link these to the .tsv.gz file that will be used for downstream analysis. So we will copy the file path, copy and paste. We will also apply some filtering criteria to reduce the noise. So min cell is equal to 10. So a peak must be detected not in less than 10 cells, and also min, and this means that every cell must have not less than 200 peaks. And now we will press the run button. This will take some time. And now ChromatinAssay object has been created. Finally, we will create the Seurat object, and for creating the Seurat object we need the metadata and the ChromatinAssay object, and both of these objects we already have generated. So now we will create the Seurat object. So create Seurat object. We will name the Seurat object as PBMC. So to create a Seurat object, we will use the create Seurat object function in R. So create Seurat object function, counts ChromatinAssay, and we will also attach the metadata with the Seurat object. So metadata, we will name the assay as a peaks. So assay. Now we will press the run button. So create Seurat object is creating, and Seurat object has been created. You can check it. Now we will look into the metadata of Seurat object. So view metadata. Now you can look at into the metadata. All the rows have the barcodes or cells, and the columns have everything from the single cell.csv file. So creating a Seurat object makes it super easy for performing all the quality control analysis later.